everyone. My name is Arjun Krishnan. I'm an associate professor in the Department of Biomedical Informatics at the University of Colorado Anschutz Medical Campus, and I'm excited to talk to you about some of the work that we do. In my lab, one of our main missions is to answer crucial biomedical questions that relate to what do genes, cells, and tissues in the body have to do with disease conditions. And the way we go about answering those questions, the thing that we actually bring to the table, is to be able to answer these questions using massive amounts of publicly available data. So, this is not data sets that we get from one of our collaborators or by being part of some large scientific consortium. This is data sets that's available to all of us all the time. So, which means you can download it, I can download it, anybody can work with it, really. And these data sets are massive because of the following reason. The massive public data sets, especially in the biomedical field, were collected over the past 25 to 30 years. These are data sets generated by hundreds of thousands of labs spread across the world. And whenever somebody generates a really good data set, they deposit this data set into a public database. Okay. So all this data is sitting out there. So you can easily imagine that because of the kind of variety of questions that scientists study, these data sets contain a treasure trove of biological signals. Unfortunately, the problem is that if anybody wanted to use these data today, it is not at all easy to identify which datasets might be relevant to their question of interest, and then also use them downstream to actually answer the question. So this is the space that my lab plays with. Okay. All right. So what are the problems with using existing data? Remember, I told you that there are hundreds of thousands of datasets, and they in together sort of constitute more than about 9 to 10 million biological samples. So this is where the data comes from. This is rich data. Okay. The first problem is that I told you, how do you find the right data sets that are relevant to your question of interest? This is typically done by looking at what is called the metadata of data. Metadata is the description about data. So whenever somebody generates a data set and submits them to any database in any field, really, they also have to describe what the data set is about. What are they studying? How was this data collected? How did they process the data? How did they process the samples? And what are some key important aspects about that particular data set or the source of the data? So they have to describe all of this in text so that somebody else can run simple text-based searches to find data sets of interest. But metadata of publicly available biomedical datasets have two major problems. Okay. The first is that metadata is highly unstructured, and second, metadata is highly incomplete. I'll tell you about each one of them in a second. What I mean by unstructured is the following. Won't it be awesome if metadata is organized like a nice table where every single aspect of a particular study is neatly organized in this table so that somebody can easily find information that is relevant to them, right? So this is not true. So what typically metadata looks like is somebody writing long sentences or short sentences, keywords, and phrases in English that includes a lot of jargon and terminology, a lot of abbreviations, and it also includes things that are potentially not relevant to finding datasets. So, for example, the names of all the authors, maybe the name of their institution, right, and so on. So we have a mess of text-based metadata that is out there, and this is what is the most common form of metadata for all publicly available datasets. So this is what I mean by highly unstructured. There is another aspect of unstructured as well. So think about two scientists who are studying a heart condition, right? One might say that we decided to generate this data set to study heart attack. Another scientist might say we generated this data set to study myocardial infarction. These two actually mean the same thing. Okay. Here is another problem. Here is another example, right? So if somebody might say, we want to study the enteric nervous system. And another person might say, we want to study the nervous system in the gut. Again, these two mean the same things. So we not only need to deal with unstructured text, but we also need to deal with the fact that people describe the same thing, but in very terminology using very unstandardized terms. So we have to deal with that as well. The second problem, like that I mentioned, is that metadata is highly incomplete. That means that if a scientist wanted to study whether diabetes impacts the molecular patterns in the body or not. They might collect data sets from individuals with and without diabetes, and when they enter the metadata during their data submission, they might only record the fact that which sample was from a patient with diabetes and which was a sample that was not from a patient with diabetes, right? That's it. But they might never tell you which tissue or part of the body they got the sample from, whether the patients were male or female, what were their age, and so on. So all this might be completely missing in the text-based description. So we have to deal with both problems, metadata being unstructured and metadata being incomplete. So over the past about 7 or 8 years, my lab has been developing a lot of AI-based technologies to solve these two problems so that once we have unstructured metadata, we can apply AI algorithms to make metadata highly structured and fill in incomplete gaps so that they become more visible. The way we do this is to apply large language models. So these are models that are very famous and pervasive right now. For example, if you've ever used ChatGPT, there's a large language model that's beneath it that's running, all the algorithms and parsing your queries and prompts and then generating results on the fly. What we do with these models is to say we teach these models how to understand biology and biomedical relationships between biological entities. So, for example, if somebody says that we studied psoriasis and they never mentioned which tissue in the body it came from, the model will know that psoriasis is a skin disease and therefore it will make a guess that the samples were probably collected from the skins of the patients. Right? So these models are super powerful. So we've been developing methods and tools and software that go around it to solve this metadata problem. So ultimately, what this gives us is an ability to make standardized high quality annotations to hundreds of millions of samples that are out there and make these annotations available for researchers so that now they can unambiguously and comprehensively search public data and find data sets that are relevant to them. So this is part number one that I want to talk about, which has to do with using AI to improve metadata to ultimately also improve data findability. The second topic that I want to talk about is might be slightly not intuitive, and I love talking about this as well, which is that if we think about biomedical research, we always think about, okay, why don't we get data from humans and then study them because we are, after all, want to benefit humans, right? Turns out that a ton of biomedical research is done using what we scientists call model organisms. These are species that are not humans, but they can be used in the lab to do very careful, controlled experiments. For example, mouse, worm, rat, zebrafish, flies, worms. So we have a ton of systems that scientists have worked out over the past 60 to 80 years, and they are like workhorses for biomedical research in the lab. Now, some of the things that we do in as scientists is that we know that every single organism has a long chain of DNA, and inside the DNA are many genes. Typically, many of these organisms have tens of thousands of genes, just like humans. And one of the things that is very easy to do in the lab is to find a gene of interest and completely delete it from its DNA and see what happens to the organism, right? So these are called knockouts. So people have been doing experiments like this and really characterizing which genes might have a role in which effect, okay, in all these model organisms. Now, if somebody wanted to study a human condition in one of these organisms, it's not at all straightforward to figure out which organism you'd use, what effect would you actually study so that it kind of closely mimics what happens in the disease, and which specific genes would you want to delete so that you can see similar effects. Okay. So this translation between humans and these model organisms is not straightforward. The reverse is also true. If you were to generate large amounts of data and insights in a model organism like a mouse or a worm or a fly, it is absolutely not clear which parts of that insight will translate back to humans, because of course humans are very complex and they are a completely different species. So a lot of it will transfer, but we don't know which parts. So these are again two sets of problems that we've been solving using AI and machine learning. A cool way to think about one possible solution to this question is that we say, again, like I said, there are about 20,000 genes in the human genome. There are tens of thousands of genes, again, in all these model organisms. Whenever we think about gene, you might hear that there is this gene for diabetes or this gene for eye color and so on. Yes, these examples like this are there, but typically genes don't work in isolation to do anything, right? Genes are like people. They work as part of a social network by working with each other. And small groups of genes work together to perform a meaningful, coherent biological function or orchestrate a meaningful process inside cells in our body. So, one of the most powerful ways of thinking about how genes go about doing their jobs is to think of genes as being nodes or entities in a large social network of genes. Just like humans could be thought of as entities in a social network of how we all operate as a society, right? Now, similar insights from society, human societies to gene societies, can be translated to make breakthroughs. For example, if you went to LinkedIn and then logged in, said, LinkedIn will give you a suggestion saying, here is your long lost middle school friend you might want to connect with. Right? How is LinkedIn able to make this prediction? Right? You two are not connected. But the way it's able to make this prediction is that your current friends and their current friends are very similar. So LinkedIn makes a prediction that you two must be connected to each other, right? So we do very similar things. We turn the same intuition into underlying mathematical algorithms, but the idea is very, very similar. So we say that if two genes share many very similar friends, they might be working with each other, right? So we make this connection and we use this to understand how genes work with each other and even make predictions about genes that we have never studied before. So, okay, now where do model algorithms come into the picture? We think about how genes work with each other, not just within humans or within mouse or within flies. We stack all these networks on top of each other so that we understand how genes correspond across species based on evolution, which has been operating over millions of years, and how genes work with each other within a species so that we can seamlessly transform or transfer information from one species to another. Okay. Let me give you a couple of examples because these model algorithms are brilliantly powerful. Okay. And especially I want to give you some, non-obvious examples. Um, we know that, for example, if you want to study how the heart cells in the humans are able to conduct electricity and how that corresponds to the heart beating correctly. Of course, there are very good models in zebrafish. This is a tiny fish that you can actually the embryo is really transparent. You can see through and see what is happening to the heart. So you can mutate a bunch of genes and see which genes might have a role in the zebrafish's ability to modulate the conductance of its heart, and that helps you find genes that might be relevant, right? So this is a case of an obvious counterpart in zebrafish. You're studying human heart condition, so zebrafish heart is a very good example, right? So we want to be able to find correspondence like this. We also want to be able to find correspondences which are not at all obvious. So, for example, one of the things that we discovered is that there is this human condition called the Bardet Beetle syndrome. The individuals with the syndrome have many issues with eyesight. They have problems with their kidney. They have problems with how their digits develop, and so on. So there are many issues that people have. So it is important to find out which model organism to use to study this particular syndrome. And based on our approach, what we found is that if you go to worms, these are like tiny wiggly things, right? Okay. If you go to worms and you see that, for example, there is this thing called ivermectin. It is an anti-helminthic drug. If anybody has problems with worms, they would go to Home Depot and buy ivermectin so that they can spray and kill all the worms. Turns out that if you actually make certain mutations in the worm, worms will become resistant to ivermectin. They won't die anymore, even if you spray them with ivermectin. Turns out that the molecular mechanisms that these genes participate in, which confer ivermectin resistance in worms, are the same genes which were mutated cause Bardet-Biedl syndrome in humans. Isn't that amazing? So, which means that you can use ivermectin resistance, the way worms develop resistance to ivermectin, as a model in the lab to study a human syndrome which you cannot study in humans, right? So these are all connections that we've been discovering. So it's been very, very exciting. Again, this is not just about a method. We want to turn this into software tools. So we've also been writing a lot of software so that other biomedical researchers can make similar discoveries. So now I've told you about two things. One is how we use AI and machine learning to solve the data accessibility and data findability problem. And the second example was how do we use AI and machine learning to transfer meaningful biological and molecular information across humans and model organisms like rat, worm, fly, and zebrafish. Right. Okay. So these are the two examples that I wanted to give you. I am very excited to be able to tell you about this in such a broad AI forum. Maybe some of the things that I want to tell you as closing thoughts is that what we are essentially doing is that we are giving biomedical researchers and the entire community the ability to mine massive amounts of existing data at scale. These data could be molecular recordings, or it could be, these social networks of genes, and whatever it is, we're giving them abilities to do this. And ultimately, what we want to do is to make sure that all these are available at the touch of a mouse on the browser. And we want researchers to have the ability to run complicated or very, sophisticated AI and machine learning algorithms at the click of a mouse, right? So which means that they can upload the data, run these algorithms, and then get back results so that they can study and plan their field. So we really want to make sure that we are able to drive this field through AI and machine learning on massive data sets. And second, of course, there is a huge mission that we have about the fact that once data sets are produced in the first place, we should not be not using them at all because they're gathering dust because they're deposited in some database. We want to keep using existing data in new creative ways to answer new questions, especially questions that even the original data generators didn't even think about. So that's my final thought, and I want to leave you there. And I'm very happy to have had this opportunity to talk to you. And I'm looking forward to any questions. Thank you.